Hello, everyone, and thank you for joining us. I'm Stetson Maldonado, the manager of the Seeds for Bees program, and I am here with Tom Johnson. He is our grower relations manager. Would you like to say hi, Tom?
Good afternoon or morning, whichever day you're watching it.
So here's our rough roadmap of how the talk's going to go today. I'm going to be going over the relationship between rhizobia and legumes, and then Tom will be taking over when it talks about in terms of the cover crop. And over here, I have a picture of a clover that I dug up, and you'll see a number of different legumes presented in this PowerPoint slide. I just wanted to point out that these will change and to notice the differences and the location of where these legumes are forming. Good grief, nodules are forming. So the star of our show is the Rhizobia bacteria. So legumes are the only plant species that can form a symbiotic relationship with Rhizobia bacteria. And this relationship allows them to fix atmospheric nitrogen into ammonia or plant available nitrogen. So the process starts with Rhizobia colonizing those roots, and then the plant and the Rhizobia will form nodules. And you can see one of those nodules on my finger, just kind of like there between my fingernail and my pointer finger. And you can see it has a bit of a unique shape, almost kind of like a rough heart. This one is on a pea plant. And there's different kinds of rhizobia, and they form different relationships, or there are different kinds of rhizobia, and some of them will colonize specific legumes, others will colonize other legumes. So there's definitely a wide variety and diversity when it comes to the relationship between rhizobia and legumes. So the reason this happens is the plants will feed and protect the rhizobia. Being out free in the soil is a little bit of a dangerous place. There's other microbes that would love to eat you. And the rhizobia, in turn, will turn gaseous nitrogen, so that's N2, into the biologically available ammonia, and that one is described as NH3. So that the NH3 is what our legumes are able to use. They can't access that gaseous nitrogen without the help of that bacteria. So here is nodulation. So inoculation is the biggest or is the most important thing when it comes to getting these bacteria to work with legumes. So there's resident microbes. So this would be microbes that are already found in your soil. And then there's the commercial products. So you could plant a untreated legume seed and it can grow and potentially form form nodules with the present or resident bacteria. If you wanted to have a more surefire colonization, you can get a commercial product. So this will either already be placed on the seed by your seed supplier, or this is something you can purchase. If you are purchasing it yourself, you do want to remember that there are different rhizobia for different legumes. So you want to make sure you're getting the correct product for what your intended plant is. Tom, did you have any notes on that?
The and then there will be directions usually on any package you might get. Most of the commercial rhizobia products come as a peat-based product. So in other words, the bacteria is grown and then basically put onto powdered peat. So it's kind of a black powder type of thing. You want to make sure when you purchase it, you're going to keep it kind of cool in the shade because it is bacteria. Packages are opaque, but still keep it cool. Check the expiration date per se on it. And then when you put it on, sometimes adding a sticker or just wetting the seed slightly so that peat will stick to it a little bit better. It might want to like flake off easier. And when you do that, try to use deionized water or non-chlorinated water. Chlorine would, of course, kill bacteria as well. So that would be another thing to be thinking of if you're applying that.
Yes, all very good points. And there's a number of mediators that will affect the size, the health, and the level of colonization of these plants. First is the presence of the rhizobia, either resident or commercial. Then there's the climate. If it's a stressful climate, the plants are going to struggle to form those nodules, and they'll have less nutrients to provide to those bacteria. There's soil type, which can affect your plant health as well, along with plant stress. Nitrogen present in the soil. A lot of people don't think about this, but if you have high levels of nitrogen in your soil, then these plants, even if you have the best of conditions for nodulation, are less likely to try to feed those bacteria. And that is because this whole process is very energy intensive, it's resource intensive. If the plants don't need to be supporting these bacteria, then they're not going to in the first place. And soil chemistry can also affect this process, specifically the pH of your soil. Tom, you were talking a little bit earlier about how chemistry could affect these.
Yeah, some legumes are more adapted to soil pHs over neutral, whereas others prefer neutral or slightly below. So, for example, alfalfa or medic would prefer to have about a 7.2, maybe even a 7.5 soil pH being alkaline, whereas peas and faba beans would actually prefer something below 7.0 on your pH scale. So the better soil chemistry for the plants you're trying to use, the better off you're going to be. And that's of course it's a range, but it's going to have some effect on how well that plant grows, and how well that plant grows is going to have an effect on how many roots there are, sites for nodulation. So it's not killing the bacteria. The bacteria is kind of like doesn't really care too much. It's how well the plant itself is going to have support for that bacteria.
Mm-hmm. And in this picture, it's the root system of a faba bean. So assessing nodule health, you can dig up your plants and see, you can pick like a few sample plants and you can dig them up and see how well they were colonized and also how healthy those nodules are. So you can, it's really interesting, even if you're not assessing it. I would say dig some up just to look at them. But you can wash off those roots, assess if there's a lot of colonization, only a little bit of colonization, and you can also cut these open. So this is the nodule of a faba bean that I cut open on my counter, and you can see how red it is on the inside, that pink red color. And the healthier those nodules are, the more red they're going to be. If they're white or kind of squishy, then those nodules were infected with a pathogen and kind of died off. What's really fascinating is that color is due to a protein that's very similar to hemoglobin called leg hemoglobin. And its job is to make sure that there is enough oxygen provided to those bacteria in order to fix that nitrogen. Something that I thought was pretty funny when I was making this slide is I was trying to remember, how am I going to remember leg hemoglobin? Is such a funky name. And then I realized it's just legume hemoglobin. So I was laughing at myself for that one.
Plant blood.
Yes, plant blood. And here you can see that there's quite a difference between species and the size of the nodules, even the shape of them. So the top one is that faba bean, the middle one is a field pea, and the last one is a snail medic. And it's kind of hard to see. I don't know if you can see my mouse, but it's right here. They weren't quite heart-shaped as opposed to just more tic-tac shaped. But you can see there's quite the difference in size comparing them and the size of the plant and the species. Don't tell my husband I was using our cutting board for dirt and roots. However, something to note, even though I have been talking about nodules so far, the nitrogen that is fixed by these nodules is transported into the top of the plant. So that top biomass is where most of your nitrogen is. It's not down in the roots. So here I'm going to talk a little bit about the nitrogen cycle. So we start off with gaseous nitrogen. The vast majority of our atmosphere is composed of gaseous nitrogen, so N2. That is taken up by the plants and transported into those nodules where the bacteria will fix it into ammonium. Ammonium is largely what is available or what the plants are able to use, that NH3. After those plants are terminated or as they decompose, that ammonium is released into the soil and it's turned into nitrites. That's the NO2 and lighter nitrates. There is a bit, it's a lot more complex than it is. It's not as simple as I'm leading it on to seem. There's a lot of back and forth and bouncing around, but this is just a very, very basic simplified version of the nitrogen cycle. And some plants can use nitrates and nitrites and nitrates. And some can produce it themselves by breaking down ammonia. It's a very complex process, and we're not going to delve into it here. But ultimately, nitrogen does return back to its gaseous form through denitrification, and it's returned to the atmosphere. So I'll toss it over to Tom for termination and incorporation of the cover crop.
Okay, well, the amount of nitrogen you're going to get out of a legume or a legume mixed cover crop is a function of biomass. So what happens is the bigger it is, the more nitrogen you're going to get out of it. The bigger it is, the more of a mess it's going to make in your orchard or your rotation. So termination and incorporation are kind of an important part of this whole process. And a lot of times it comes down to timing and how you're going to do it. So the timing, if you want to get kind of that sweet spot of maximum biomass and residue size you're able to deal with, you're probably looking at your cover crop, and shortly after it starts to flower or up to about 20% flowering, you're still not filling seed pods and the plant's not getting very tough. It's going to break down. It's not necessarily the plant concentration of nitrogen is probably about as high as it's going to get. It's just how much more weight you might put on if you let it go longer. So you're kind of going in that point, so that starting flowering to about 20% blooms in the springs, usually around 10 days to 14 days after you start seeing flowers, it has pretty much hit that point. So then what you're going to do, whether you're going to just mow it or you're going to incorporate that biomass, is going to have something to do with basically how well it breaks down and how much nitrogen and how fast that nitrogen is going to be available into the soil system. So mowing it just by itself is going to leave that biomass on the soil surface. In the grand scheme of things, we could probably expect about 40% of the plant nitrogen to become available in the soil in that first year. Might be a little slower due to its being slower to process. There's less biology breaking that plant material down, but that's a pretty decent rule of thumb. It's about 40% of what was there you're going to recover. If you incorporate it, you can get probably 10 to 15% more of that nitrogen available in the first year. The difference, of course, basically is how old your trees are and your other management systems going on type of thing as to what you would do. Bearing orchards are usually not going to be incorporating it, so leaving it on the surface is going to, one, slow down how fast that goes back into the system, and also probably ding you a little bit on your overall amount you get. But on the other hand, you're leaving your harvest bed where it needs to be.
Yeah, and then some of this nitrogen will show up in the second year or sometimes even the third year as it continues to be released.
Yeah, the breakdown and release of that nitrogen is a biological system. So it's a lot different than going out and putting a manufactured fertilizer on that breaks down with pretty predictable results. Using this biological system basically is not as predictable in timing and intensity, but on the other hand, they've studied it long enough. They've got a real good handle on how much you actually do recover out of that. So then you've got a cover crop out there, and it may be one of those boxes you checked on your nitrogen plan that the water board or someone may want you to have to turn in type of thing, and how much nitrogen you're going to apply or how much you plan to apply or how much you did apply, that type of stuff. So a cover crop is potentially a contributor of soil nitrate, soil nitrogen type of thing. So there are ways to calculate how much nitrogen you're going to get out of your cover crop. One of the methods is going out and cutting a 2 foot by 2 foot square. And of course, you want to do this in more than one location in that orchard because of the variability going on, and you kind of average all your numbers. But you're going to take all that plant matter within that 2 by 2 area and dry it, and weigh it late once it's dried down where it's not drying down anymore. You can actually do that in a microwave. It's just you got to do it slow and easy. You could put it in the oven. You put it in paper bags and let it sit out in the barn and dry slowly. That type of stuff. It's usually not like you have to have an instant answer anyways. You're going to take that dry weight and you're going to times it by 43,560, which is the square feet in an acre, divided by the sample area in square feet, which is teeny tiny. That's going to equal your total dry weight per acre. It's going to be an estimate even though you took 6 samples out of it. It's still going to be kind of an estimate. Then you're going to take that same as above, but if you had small grains and legumes in there, when you sampled it, you could dry all of it, or you could pick all the nitrate, all the legume materials out of your sample and separate them out and just dry them, because that was kind of what we're after. We're just after the nitrogen coming back from that thing. So you could do that, separate it out, dry it, weigh it, do the same multiplication and estimation math on that. And then you multiply that dry weight per acre by the following. So if you went out there early flowering, in other words, you just saw a few flowers out there, you're probably going to hit that number with about 4%. If it was pod forming or starting to fill seeds, because legumes usually make seeds in pods, groups of seeds in a pod like peas, like snap peas or whatever. So if it was starting to form pods, you're probably going to hit that number with about a 3% factor, for the mere reason that, the plants were heavier, but at the same time, the plant chemistry was starting to shift a little bit. If you kept the grasses out of that thing, weighed them and all that fun stuff, and you dried that down, you're going to use about a 3% factor on what that weighed in the dry matter. You could also kind of guesstimate, when you took the sample, it's like, it was 80, the biomass in this sample was about 80% legume today, or it was 60% grass, or whatever it was. And again, that pod forming non-legume, we're going down in percentage that we're going to use on our factor, because again, the plants were getting a little bit older, a little bit more stiff and stem, and less overall nitrogen in them. And then basically you're gonna you can also kind of guesstimate this type of stuff if you want. Average nitrogen ranges. In other words, what they did was they harvested the top growth and did these calculations to it. They may even done lab work on it, and they may have checked the soil as well. But if you had a good stand of winter peas and you got them up to flowering pretty good, you could get between 20 and 90 pounds of nitrogen out of that cover crop per acre. Red clover, a lot more variable, but I would say 50 to 100 pounds is probably a little bit more on the realistic side. And the rose clover, 50 to 75 pounds. And part of that's just based on plant mass that I'm familiar with. Subterranean clover, kind of in the same boat, 25 to 175 pounds is potential. Hairy vetch, 50 to 150 pounds. A good stand of hairy vetch that's blooming is probably going to be close to 100 pounds of nitrogen out of that biomass. Crimson clover, pretty similar to red clover. Bird's foot trefoil, in our climate, is a perennial. So if you went out and mowed it while it had flowers on it and let that slowly break down out of there, you're probably looking at about 50 pounds of nitrogen, but it's going to grow back. So the next time you mow it, it's going to pump a little bit more into it and a little bit more into it, that type of thing. So it may be more cumulative effect. We want to say these numbers came from the University of Georgia. So again, their climate's going to be a little bit different. But this has been watched and studied quite a bit. So we've got a good handle. These numbers are, in my opinion, well within the range of those things' capability.
Yeah, and I just want to point out and remind everybody that this is very much tied to the above ground biomass. So if you're wanting to hit these higher numbers, you're looking at an incredible amount of top growth, just to keep that in mind.
So then we're doing nitrogen plans because we're required to, but at the same time, there's an economic reason behind it. We kind of want to know what that's going to cost us every year. So the cash crop requirements, and this is just basic replacement for the leaves and the crop that's taken off, not necessarily for growth or whatever, but almonds is going to be somewhere between 85 and 110 pounds per acre, at 88 to 108 pounds per acre. In vineyards, you're probably looking at 30 to 70 pounds. My rule of thumb was always about 40, 45 pounds of nitrogen for vineyard growth. Walnuts, bigger root system, bigger trees, a lot more big leaves. So yeah, we have a higher requirement, 150 to 200 pounds of replacement nitrogen per acre to be put on there. And that's kind of an average of those replacement applications for mature orchards and vineyards. Younger ones are going to be different. Reduced crops and things like that are also going to fall in that place. So that's kind of that gives you an idea. Okay, I'm going to, just for trees and crop on my almonds, I'm going to need 100 pounds of nitrogen at least. And that doesn't take into account growth. So if we take that cash crop requirement, so our almonds are going to need about 100 pounds. And if we got a really good stand of hairy vetch out there, we could make that amount of nitrogen. Not necessarily all going to happen this year, because you got to remember we're looking at 40 to 50% of that's going to be available that year. But the hairy vetch, if you went and cut the tops off and weighed it and everything else, yeah, that 150 pounds of nitrogen is potentially there. So again, we're going to mow it. Okay, so we're going to have 40%. So we're going to get about somewhere between 20 and 60 pounds that first year. There'll be a little more shows up over time, but we're not replacing applied for fertility. If we incorporate it, yeah, we're going to get a little bit more. We're going to get that 10% jump in what would be in the soil and the trees could get to this year. But basically, we're not, like I said, we're looking at half or less of what that cover crop could produce. So again, those trees are going to need X amount. We're not necessarily going to be able to replace all of it with a cover crop, but we're going to make a dent in what it has to have. And then, of course, I'm sure there's more questions than there was answers to this thing. So we kind of skimmed some of the really in-depth things for so on and so forth. But we're willing to answer questions and all that fun stuff if at all possible. But we want to thank our donors, Seeds for Bees, which we have a cover crop that has legumes in it. We can offer other ones. We can also technically advise you on ones that have that in that. But Seeds for Bees is entirely funded by donations from individuals and corporations. This makes this program stackable with both federal and state grants. They don't interfere with that type of stuff. We're grateful to the people who make our program possible. Many of them who have been doing Seeds for Bees since it started, veteran cover croppers, and they will also tell you that, yeah, it's not straightforward, but it's there. So you could use this QR code to donate, or you can find us with a Google search of Seeds for Bees or even Project APISM. Thank you very much for being here today.
Thank you. And I hope you all have a wonderful rest of your day.